Okay, thanks for coming. Good morning to everyone. Today we host Ramón Perea. He's coming from the Universidad Politécnica de Madrid as a titular professor, and he did 10 years ago, more or less, the postdoc here with also seed dispersals. And well, he's going to do a little bit of presentation, so you can start. Oh, no, you have
I have this microphone.
Exactly.
Thank you.
You're welcome.
Thank you, Maria.
Thank
you, everyone, for coming, for the opportunity to show our results here. It's a real pleasure to come back 12 or 13 years ago when I was here with Kani and Miguel doing some research on seed dispersal. And it's always a pleasure to come back to Seville. And I hope you like what I'm going to present, which are two research projects that are right now, and we are almost done with them. First of all, I would like to introduce who we are. We are a recently created group called Paella, Plant and Animal Ecology Lab. And we are working from Madrid, in the Universidad Politécnica de Madrid. And as you see here, we work with animals, but we also work with plants, and mostly with the interaction between both. So it's mostly animal ecology and conservation. Our main group are the cervids, the wild ungulates, but we also work with raptors and the interaction with these wild herbivores. For instance, with this equilibrium project, looking at how the golden eagles are predating upon the roe deer fawns. We're also working with the effects of elephants in Tanzania, mostly on the croplands and the conflicts between humans and elephant populations. And from the plant perspective, we work mostly on the quercus, on the oak regeneration cycle, plant facilitation, also the pastures and the effects of herbivores on the pastures, and the restoration projects. And the main lines I'm going to present today are dealing with plant-animal interaction, mostly herbivory, seed dispersal, and animal-soil plant interactions, and also working on the ecological indicators for sustainable populations. So this is the new group from Madrid. We have a very applied component in our research, so we are interested in implications for management, and this is also one of our main lines there. These are the main ingredients of the Paella lab. So now there are many pre-doc, some postdocs too. We have lots of collaboration with other institutions. So what I'm going to present today is the two projects, as I said. The first one, both of them are dealing with ungulates. What are the ungulates? Ungulates are this clade of vertebrates that have hooves. And they're mostly herbivores, but there are some omnivores, like the wild boar. And the main ungulates that you may think of are these large herbivores here: deer, roe deer, fallow deer, mouflon, chamois, Barbary sheep. Those are the ones that we are mostly working with. And they are considered ecosystem engineers because they play a very important ecological role in the system, as you can imagine. They provide landscape heterogeneity because they maintain different patches of vegetation, right? They also play important ecosystem services. For instance, they also reduce the fuel, and they prevent, or they can prevent, wildfires. They are very important also in conservation of protected habitats that we are obliged to preserve, for instance, from the Habitats Directive, the Natura 2000 Habitats Directive, because they somehow cause these top-down effects, but also bottom-up effects on the system by changing the composition of the plants, of the herbaceous vegetation, of the woody communities. Also, they are very good at dispersing some seeds, but they also prey upon other seeds, so this is a very important role in the ecosystem. And they also play a very fundamental role in the food web by supporting the communities of large predators and also decomposers, right? So, what's the situation? Over the last decades, we have an increase in wild ungulate population in Europe, in North America, and also in Spain. And there are many different reasons for that. One is the current game management, mostly in southern Spain. You know that there is these hunting states where they are somehow favoring these populations by using fences and promoting wild boar and deer mostly because they are this hunting interest. And there are these densities, as you can see here, above 70 individuals per square kilometer, or wild boar above 20 individuals per square kilometer, when naturally should be about 10 or less than 10. Additionally, there is a land use change. As you know, there is this rural abandonment, and this is favoring the increase of forest land. The livestock is decreasing, so sheep, goats, and so on are decreasing, and they are somehow regaining this space that is now increasing the forest land. There is also a lack of predators, of course, in many areas. So the wolves, and which is the main predator, is not able to somehow prey upon these communities. And also we have had many new reintroductions or introductions of species that with no planning behind that. So the populations have found a new space, they increase exponentially, and now there are some problems with the management. This is, for instance, in Guadarrama National Park, where there are populations above 100 individuals per square kilometer, when in other areas we find about 15 individuals per square kilometer. So this is the situation over the last decades, as I said. Porter already indicated that in the northern hemisphere, we probably have the highest population of wild ungulates ever. What's the situation in Spain? In Spain, we have these ungulates, as you see here, the main ungulates that you all probably know. This is how they are expanding from the 1950 until right now. So if you look 70 years ago, the deer were only in these southern part areas. Most of the deer are coming from, or the deer from the northern parts are coming from the southern part that were reintroduced. And this is only, you can see here how they expand, but also how they have increased the population of wild boar, red deer, and other ungulates over the last 50 years. So this is the current situation. So this has caused a main change, as you can imagine. The first question that many managers from national parks ask is, but what is the natural population? What should we expect? And this is very difficult to answer. So we did a study on paleoecology, using paleoecological records, using this course that you introduce in peatlands, where the materials can be preserved for very long times. And there is a kind of fungus that only grows on large herbivore excrements. And this is a good indicator. This is the one you see here, is the spores. We have found the spores of this dank fungal organism along the last 4,000 years in Cabañeros National Park. This is what you see here, that the abundance of these spores is much higher, about 4 times higher, 4-fold higher than the last centuries here. In addition, there is another organism, sphagnum, which is a moss growing on these peatlands, that is very sensitive to herbivore pressure. And we have found the opposite, a reduction of this moss over the last centuries, which indicating that probably we are having 4 or 5 times higher densities of wild ungulates in comparison to the last 4,000 years. This is just to give you an idea. So then we came up with this project that we call HEREGE. HEREGE is herbivory and regeneration. And HEREGE project tried to evaluate the effects on the system, mostly on the vegetation and on the soil. And try to search for reliable ecological indicators, because the problem is how many animals should we have in every national park. So the thing is, we're going to look for ecological indicators to estimate these sustainable densities. But we want to try to also to analyze the context dependence and try to map the pressure, the herbivore pressure, so that the managers of each national park have an idea which areas should be prioritized, how they should manage the populations. The idea is to somehow establish a long-term sustainable management, looking at ecologically sustainable population densities, because this is a national parks, we have to preserve the ecological values. What are the main areas of conservation? What species or habitats are more sensitive to these wild ungulates? And what are the main areas for restoration, for favoring regeneration, natural regeneration? HEREGE project has been focused on 3 national parks, Cabañeros, Monfragüe, and Doñana. They are dominated by these wild ungulates, as you see. Doñana is peculiar because it has also fallow deer populations. And the vegetation is very similar. There are scrublands, Mediterranean shrublands, evergreen shrublands, right? So we started to look at the effects on vegetation and soil on this national park, and we established 226 plots. These plots are 10 meters diameters. This is the distribution in each national park. In Cabañeros and Monfragüe, we only did it in public land. We could not go into the private land. But for Doñana, we were focusing only on woody vegetation. Why are we focusing only on woody vegetation? Now we will see on woody vegetation. Because woody plants are the ones that are more sensitive to herbivores. Some herbaceous plants benefit from herbivory, and the woody plants have no such good mechanisms or adaptation to high levels of herbivory pressure in comparison to herbaceous plants, which are also annual plants, more difficult to follow, more difficult to monitor, and so on. So for each of these plots, we measure woody plant cover, plant height to estimate the fetal volume, the browsing degree, the browsing impact, the deer pellet groups, the number of pellets of deer. This is to estimate the habitat use and the relative density. It does not tell us the number of individuals we have, but it can tell us the relative density and the distribution in different areas. The rubbing and trampling damage that they may cause, the wild boar rooting activity by measuring the area affected by wild boar, and also the natural regeneration. Counting the plants, the natural regeneration, every single plant that we could find there in this transect. And for those, we measure the height, the basal diameter, the relationship between the height and the basal diameter. This is what we call slenderness coefficient, because it's a good attribute of as we will see, of herbivory pressure. And we were also very interested in the microsite, where the regeneration were occurring, to give some idea, to give some management implications to favor the regeneration under those preferred microsites. Here you see what we have been using is the browsing degree. This goes from 0 to 5, is a semi-quantitative value, and then you see that the slenderness coefficient, which is the height divided by the diameter, it's decreasing as herbivory pressure increases. So this is some examples of plants with very high herbivory level. Some of these, and this is Erica scoparia. Some of this plant is highly rejected, so it doesn't go beyond score 3. And the thing is that what is the sustainable level of pressure for each particular species? We consider the sustainable pressure when it's beyond 3. Why beyond 3? Because beyond 3, so 4 and 5, we couldn't find any flowering, any reproductive structures. So there is no sexual reproduction. That's what we consider the sustainable threshold. If the plant cannot sexually regenerate, then the level of pressure is unsustainable for that particular species. This is just how we estimated the scores, looking at the proportion of branches that are browsed, and the shape and the lack of flowering structures on the levels 4 and 5, as you see. But it is not the only effect they cause. Other effects are also the rubbing with the antlers. They have the antlers and they shed the antlers every year. They are going to rub against the trees, and this is a Pistacia terebinthus killed by a deer antler. Why? Because they also prefer some plants over others to eat, but they also prefer some plants over others to rub their antlers. And they prefer these ones because they have aromatic compounds. Pistacia has cementine, aromatic compounds, resins, and they are looking for these particular trees. And after many individuals rubbing in many years, they are able to kill some of the plants. We estimate the damage also by looking at these semi-quantitative values, similarly to how we did with the browsing, but looking at the circumference of the tree. What was the proportion of the circumference rubbed? If the 100% or almost 100% of the circumference is rubbed, they girdle the tree, they kill the tree because there is no flow of sap, right? After that, we estimated the preference of deer over all the species that we found in the 3 national parks. This is consumption, this is browsing, And then we find very similar values. These are the ones that are preferred, and this is the ones that are mostly rejected in terms of browsing. They reject many conifer species, toxic species, the ones that have a lot of secondary metabolites, and they prefer this one that you see here, Pyrrhus, Adenocarpus, Filerea, Olea, all those species that you also have here in Doñana. For Doñana, we have also this list, Myrtus, Osiris, Genista anglica. All these species are the ones that are more preferred in terms of browsing. You have here some pictures of these Filerea, for instance, heavily browsed, or Myrtus, or there's no production of flowering here, Erica scoparia, Juniperus, which is one heavily rejected in terms of browsing. So what we were interested in, now what do we do? Are we going to focus on all species? We're going to look at all species in all national parks. I said this is impossible for a manager. We're going to look for species indicators. Let's look for species indicators. So we did that with several species, and then we found that the ones that are rejected, even though the number of deer droppings, the pressure, the herbivory pressure is increasing, the level of this browsing score never reaches the threshold, the level 3, right? Level 3 is what we say the sustainability threshold, where there is no production of flowers or fruits. And even though there were increasing number of deer droppings, this species didn't work well. Why? Because this is highly rejected. Doesn't matter how many individuals we have, that is going to be quite heavily rejected. On the other side, so in contrast, we have the opposite, a Myrtus communis, which is heavily preferred species. And even though we have a low number of deer droppings, the browsing score is above the sustainable level. So we were looking for something intermediate that would have the slope is quite high, so it's very sensitive to herbivory pressure, and it goes from 0 to 5. These examples are Erica scoparia, Filerea angustifolia. These are what we propose to use as species indicators. Now what we did is, okay, we're going to focus on these species as indicator because they are widely distributed. This is very important. An ecological indicator has to be easy to measure. If it is difficult to find, this is not going to be a good indicator. So these species are widely distributed in the 3 national parks, in this case Filerea angustifolia, and we were looking at the browsing damage on the different areas, and we somehow built this map. The red and orange colors are indicating 4 and 5 herbivory level, so unsustainable level in all these areas of Cabañeros National Park, which is about 58% of the area is showing unsustainable levels of herbivory pressure. This is also this is only for the public areas that we did, and the same with Monfragüe National Park. About 60% of the land is showing unsustainable levels of herbivory pressure. And for the case of Doñana, we have even a little bit higher, about 81% of the area is showing unsustainable levels of herbivory pressure, as you see. These are only the ones that we found, Filerea angustifolia, the dots that you see there. There are many more plots. This, together with the deer droppings that we found in each of the plots, here you have the habitat used by deer, looking at the deer droppings in Monfragüe, Cabañeros, and Doñana. Using these 2 variables, the level of Filerea angustifolia herbivory pressure and the deer droppings, we established a relationship. Why are we establishing this relationship? Because we want to know how many animals could we somehow have to obtain the sustainable levels. So this relationship is showing here the browsing score from 0 to 5 and the number of pellet group of deer pellet groups per plot. If we look at the sustainable threshold, which is 3, we should have about 6 pellet groups per plot. A plot is 78 square kilometers. We should have that value, approximately, after doing many replicates of many plots. But we have, on average, what we found is 8.06. We should go from 8 to 6, which means that the population must be reduced by about 1/4, so 25%. This is what we did with Cabañeros. We have done also with Monfragüe, and we are also doing with Doñana. And with Doñana, the average number is 8.3 pellets group per plot, and we should have about 4.8, which means a reduction by 42% of the population. What is the last census of deer here in Doñana? The last census was 2,200 individuals of red deer, and there should be about 1,275, according to this. And this is what you have here is the dynamics of deer population over the last years. Here is only national park, but from 2010 onwards is natural and national park. So we can only look at this part of the graph. And this is how it has increased the deer numbers over the last years. And then according to this, it should be like it was in year 2010, approximately, okay? This is interesting for the managers to know what should be the sustainable populations of deer. But for wild boar, it's more complicated. We have not found a sustainability threshold to say that there is a level of sustainability. And we were looking at the effects on the ground by looking at the rooting activity. I'm mapping this rooting activity in the 3 national parks. This is the area affected by rooting activity by wild boar in each national park. So giving us an idea of what are the areas more affected. And looking at sustainable parameters, indicators, we thought that looking at the protected habitats, which we are obliged to protect by law, because this is directed habitats, we have to protect this kind of habitats of communitary interest. In Europe, you have here the code. We were looking at the most sensitive, the most affected habitats. And then you see some are of priority conservation, the ones that you see here, like the dunes and like the cladium peatlands, which are very sensitive to this kind of changes. And then you have also Salicornia pioneer communities, you have the grassland, the humid grassland communities, the Quercus suber communities. These are the most sensitive habitats that managers should focus on when trying to estimate the wild boar population or wild boar effect. And the problem comes that with many other ungulates, maybe they are not growing on woody plant communities. And how should we deal with this? For instance, with the wild goat that you have here, Capra pyrenaica, this is, they are somehow inhabiting these rocky areas, and there are no woody plants, or very little, and the ones that are like this, right? So we thought that maybe looking at the rocks and looking at the soils, it would be a good indicator for this kind of wild ungulates. And this is what we did, because you see here, they are eating the lichens and the mosses of the rocks, and they are also trampling on them. So we thought, okay, let's look here for a new indicator based on what we call the biocrust, which is this composition of these communities made of mosses and lichens. And we were looking at the effects of wild goats on this. Here you have low herbivory pressure by wild goats. You see the moss cover here. And then here you see when the pressure is increasing, how these mosses are decreasing somehow, these patches, right? So we also relate the moss loss with the drop in abundance for, this is Guadarrama National Park, to estimate how changes in the drop in abundance are affecting to the moss loss. And which is also very important is that we are noticing erosion problems because of this removal of the cover on the rocks. And here you see this band. This band it should be with lichens, and then you see there is no lichens because the soil was here, and then it's been removed, and you see all this loss of soil from this. So we are measuring with the help of some geologists, measuring the loss of soil. You can imagine that the loss of soil is a very important problem because the recovery of soil, it takes centuries, millennia. And then if you lose the soil, then you lose the whole structure of the ecosystem. And this is a very important and relevant indicator. So the second part of the HEREGE project was going a little bit beyond what we say the herbivory and the biotic pressure, and we were focusing also on the joint effect of biotic, herbivory, and abiotic stress, caused mainly by climate change, by the increase of intensity and duration of the droughts. And then we were focusing also on restoration activities to try to somehow recover those areas that are not regenerating properly. What did we do? We planted seedlings of Quercus. Quercus suber and Quercus ilex, depending on the national park. We planted a total of about 1,200 seedlings. And then we performed several treatments. We had protected from herbivory, no protected from herbivory, under shrubs or in open areas. We also removed the acorn, or they staved the acorn, because looking at the wild boar effect on acorns. We also cut the above ground biomass, or we left it as it was, or we also watered the plants, or we did no watering on the plants. Different treatments, okay? Lots of treatments here, few treatments here, with the idea of looking at what treatments could benefit or could somehow favor the regeneration of these oaks. Here in Doñana, you have lots of plantations that they are doing with Quercus suber, and in other national parks are doing the same, but they want to know also how they could improve this, what areas are the best, but what treatment are working best. These are some of the damages of the impact that we see. This is deer on Quercus seedlings, pathogens, rabbit, because it's a different cut, it's like a very clean cut. Wild boar rooting activity, they like a lot of going to the seedlings, they find them very easily, they remove the acorn and they eat them, and also the drought, which is one of the main causes of seedling death. What we found is that the type of damage changes over time. And it's as expected. We were expecting a heavy drought damage along the summer. So particularly from May onwards, you see that the probability of drought stress is very high. But what we found is the damage by wild boar was really, really heavy at the beginning of plantation, which is that they are done in January, February in most areas. And there was a heavy attack of wild boars on these seedlings. And then in May, June, when the grass dries off, deer, rabbit, and so on will start to browse on the seedlings. But the main damage at the beginning is caused by the wild boars. This is what we found by looking at the survival probability of plants, of seedlings, along the growth period. And you see that the 2-year seedling plants, because we were planting 2-year-old seedlings versus 1-year-old seedlings, 2-year-old seedlings showed greater survival, so we recommend to use 2-year-old seedlings in this case, because the 1-year-old seedlings are heavily affected by wild boar, but particularly those that have the acorn attached. If you remove the acorn from the seedlings, at least what we found in Doñana, you increase the survival of these plants. Because wild boar will focus in on removing, on root out the whole plant and eat the acorn. The thing is that the 2-year-old seedlings suffer more drought, so they are better for wild boar attack, but worse for drought. So what should we do? Then we thought, okay, how can we deal with this drought condition? How could we reduce this effect? We thought, okay, we could use watering, which is very artificial, as you can imagine, but this is what they are doing in many national parks. They water the plants for the first 2 summers, and it works. But also we wanted to compare this, if we cut the above ground part of the seedling, how are they going to respond? Okay? Because we know that they are able to resprout, right? So we wanted to see the effects of this, and we found that the cutting, this is the survival of the seedlings, we are doing it in 2 national parks now. We found that the cutting the above ground biomass has the same effect as watering the plants, because they survive very well when they cut them. And the thing is that what we are finding now is that if you cut the above ground part, the deer will not see the plant there. So you are also somehow reducing the first year effect on this survival of plants. But the other part is that if they re-sprout many times, the new sprout is lower and not so vigorous as the first one. So this is also somehow counterbalanced. But we found a compensatory effect of herbivory by removing the above ground biomass against drought. And this is a good possibility of using this as a strategy instead of watering the plants. Another interesting aspect is that we're using these protectors and we're using nursery shrubs, shrubs that cover the ceilings. Shrubs are cistus or Alimium alimifolium here in Doñana to protect the ceilings. So we planted ceilings underneath the Alimium and cistus and looked how they perform ecophysiologically and survival. We look at both. And then we found that under shrubs, the physiology, the parameters like chlorophyll are better, so they are performing better under these conditions. We found that particularly this effect is particularly important in areas where the plants were fenced. Why where the plants are fenced? Because where the plants are fenced, they have this dry herbaceous biomass. The herbaceous biomass is competing heavily with the ceilings. And then the effect of this plant facilitation on the ceilings is much better in this fenced area than in open areas like you see here where there is almost no herbaceous plants because deer are all the time grazing in this area. So the net plant facilitation was greater in these areas where herbaceous biomass is abundant in comparison to these ones where they are not so abundant. Okay, I would like now to present a project, what we call Incremental project. This is not done in any national park, this is done in 2 different areas. But this is a project funded by the Ministry of Science. And we call it Incremental because we are looking at the effect of incrementing deer populations, wild ungulate numbers, on enclosures, as you will see now. So what is the main hypothesis of this project? The main hypothesis is that changes in wild ungulate populations are going to cause significant changes in the structure, in the composition, and in the functioning of ecosystems, but also in the provision of important ecosystem services like wildfire prevention, the fertility in the soils, health, and so on. So this was a very ambitious idea, as you see here in this graph, but trying to look with an integrative approach how these increasing densities of deer affect different biodiversity components from plants, vertebrates, invertebrates, but also looking at the effects on soil, microbes, microbiomes, looking at the interactions between plants and animals, we will see now, pollinators and for instance, and plants, and also looking at the different ecosystem services. So the novelty of this project is the approach covering the whole ecosystem. The predictions that we had is that each component of the system, here you have soil microbiome, invertebrates or plants, whichever you think of, each component and each process, interaction for instance, will respond in a different way depending on the abundance of deer, low, medium or high, but also on the time that has elapsed since the start of the pressure. This is the time here, because we have introduced different densities, and then it's not the same if you have been there for 1 month that a deer have been there for years, right? So this is the main prediction. This is just hypothetically. Then we establish enclosures, and we introduce deer at different populations. This is low, medium, high, and very high deer densities. And we used 2 contrasted systems, Mediterranean systems, but they are different in terms of the soil characteristics. Quintos de Mora in Toledo has acidic soils, perennial vegetation, mostly dominated by evergreen, but also oaks. And then Muela de Cortes, which is calcareous, very basic, very different soils, but also dominated by woody plant communities. This is a coordinated project that we started 4 years ago, coordinated with the Universidad Autónoma de Barcelona, working with veterinary aspect mostly, because they were also interested in the animal health and the animal welfare, because we were introducing new animals there. So they're looking at the performance of the animals, their reproduction parameters, their health, how they compete between each other, and how stressed they are. And then from Politécnica de Madrid, we were interested mostly in the ecology part of plant and soil, looking at the plant diversity, what are the resources for the animals, what we call food escape. These are the map of the resources for the animals, and the effects on invertebrates, pollination, plant physiology, soil properties, and so on. These are the main variables that we measure. This is structured in 3 main groups: the biodiversity components, plant diversity, vertebrates with phototrapping, invertebrates with pitfalls, parasites and ticks, soil microbiome, then processes and interactions, like animal competition in terms of deer competing with each other, interaction networks, trophic efficiency of the system, herbivory and preference and regeneration indexes, physiology of the plants, and then the health and ecosystem services group, which is how are this increasing of deer are affecting the overall health, not only the deer health, but also the human health, because some of these diseases, maybe you have heard of, can also be transmitted to humans, what we call zoonosis, like tuberculosis or the fever, Crimea-Congo fever. There are other diseases that are important for humans. And also the carbon sequestration and emission, the soil, animal health, as I said, fire risk. We're looking at how deer are changing the structure of the vegetation and the flammability of the plants, and conservation of protected species and habitats, fertility of the soil, and so on. So as you can imagine, this is a very ambitious project to be done in 4 years, and then we involve many other institutions, foreign institutions or national institutions. And we are really grateful we are very grateful because we were searching for experts on every different topic. What we did in the first year, it was the pandemic, it was a little bit crazy. We measured the reference level of the system before we introduced the animals, and that was done in 2020, sorry. So we were measuring just to obtain the reference parameters, and then we introduced the animals at different densities, very high and medium and high, and then we had the control plot, which was browsed, for the last 20 years. And then last year, after being 1.5 years in the enclosures, we took out the animals, and we are looking now at the recovery of the system too, how the parameters we have been measuring, they are able to recover or not. So we're looking also at the resilience of the system. As you can imagine, this is a very heavy impact, but in very short time. So the idea is to look at other exclosures that we have in the national park, in Cabañeros and in Quintos de Mora, to look at the long-term effect, because this is a very intense but very short effect, as you can imagine here. These are the parameters that are measured for each animal that is introduced, the biometry, there is blood analysis, feces analysis, and so on. They are marked, and then they are released. Okay, some of the preliminary results, because we are analyzing now the data we have collected, is, for instance, we have 11,000 individuals of arthropods to be analyzed yet, and we are looking at the size spectrum of these communities, invertebrates. The invertebrates were captured here with the pitfalls, and then we are scanning to estimate the biomass of in each of the treatments, and with that we can estimate what we call the trophic efficiency by estimating the size spectra. This is based on the assumption that the largest invertebrates will prey upon the small ones, right? And then we can estimate this trophic efficiency. Some of the results we have found is a reduction of feet of volume. That was something expected, right? The reduction of of biomass in the different, as deer population increase. Changes in plant flammability, because these, some species are almost killed, like this one, and this is a rejected one, supposedly. And then what we have found is that there is much more dead material in the heavy browsed areas, causing an increase in flammability of the plants. We are doing that with the INEA fire lab. We also found an increase in some compounds, for instance, anthocyanins, so it's a good estimator, this secondary metabolite is a good estimator of an increase in the herbivory pressure. We are finding that the functional diversity is somehow homogenizing as we increase the deer density. For soils, we are not having great differences, so it seems that it needs probably longer time. We have only found differences in nitrogen, but not in carbon or apparent density or other variables. And the microbiome, we are still analyzing this, so we have no idea. One interesting result that we are finding now is the simplification of these plant-pollinator networks as the deer density increases. As you see here, this is mostly done by Hernandez Castellano, who is leading this manuscript. And you see the simplification of the plant-pollinator communities. Some of these plants almost disappear. For instance, crepes, campanula, and other plants disappear from this. So we have less modules here at very high density, simplification of the system as we increase with the deer density. Another interesting aspect that we are focusing on is the spectral response of plants. We are looking at these using different devices, mostly drones, but also we have the field spectrometer and also satellites. We are looking at the response of plants spectrally when they are subjected to different stressors, herbivory stress and drought stress. And we are able to identify plants with these spectral images, so we know which ones are Quercus crucifera, which ones are Quercus ilex, and so on. And we are able to build these food escapes. So this is a map of resources for our herbivores. And we are also looking at the phenological changes. You can imagine this is very different if you do this in spring than if you do it in late summer. And these are some of the main results. What we are using here is what I said, field portable spectroradiometer, because this is, you can get much more detail. It's you just do it on the plant directly, not using drones or satellites. So we have sampled 397 individuals of these 6 species. And with this device, you can obtain 512 spectral bands. So we are obtaining different spectral indices, looking at the sensitivity to herbivory, and looking also at how it depends on the palatability of the plants. We have highly palatable plants, but also we have very highly rejected plants, and looking at how it changes this spectral signature with the stress. Some examples are this index that is very popular, NDVI, you may have heard of, how it changes with the browsing degree, and how particularly above level 3, which is the sustainable level we said there, it's much more sensitive. Other spectral indices, like the photochemical here reflectance index, which is related to the light use efficiency, is not so sensitive to deer browsing. With that, I would like to finish thanking all these people and funding from the National Parks Agency, from the Ministry of Science, and from the different programs and all these institutions, and many people, so I don't have room here to write all the names. And I would really happy if you have any questions or comments. Thank you. I think you need to turn it on, right? Yes.
Super interesting, thank you. I really enjoy seeing the plant-pollinator networks, because when you started talking about which plants are preferred by deer, I was thinking those are the same plants that are preferred by pollinators. But my question is about like within a species competition. So do you see a signal of self-regulation? So when they overgraze, they reduce their populations because they are competing among themselves, or this is not a process?
You're talking about plants or pollinators?
No, so sorry, we're talking about deers.
How they compete?
So deers can compete among themselves, so they self-regulate their populations when they overgraze the resources?
So we have, I didn't say that, we have been using female individuals of the same age, 1.5 age, so young individuals, and they compete. And when we put them together, some were really fighting at the beginning. So it was competition, but when they had lots of resources. And then what we have found is this competition for resources. You mean if they regulate the population of deer? We have done this in 1.5 years, and we have no data on that. But naturally, they would what other studies have found is the reduction of the performance of the animal. So they first reduce in the males, they reduce the undersize. In the females, the survival of the fawns, and then the fecundity of the animals. So the first thing you notice in a population of animals when they increase is this reduction on the survival of the fawns, and then the reproduction of the fecundity. Some deer can be already, they can give birth animals at the age of 1-2 years, but if their resources are very low, they will wait for 1 more year until they reach the certain weight. So, and some years, in very dry years, they lose their fawn, and there's one missing year until they recover the body condition. And when they recover the body condition, they can produce new fawns. But yeah, that's interesting. Yeah.
Hi, thank you very much. It was a really interesting presentation. I agree with Nacho. I think maybe a bit in line with self-regulation, what Nacho was saying, but then on the plant side. So because you measured the suitability or the sustainability index based on how many flowers are browsed away, and you know, they're shortening, but because there may be some feedbacks generated, right? So plants that don't produce flowers the 1 year actually may grow better in the next year, or the few seedlings that they have, you know, they have a better, you know, like, did you look at this kind of potential for the plants to actually do better than expected because of, you know, and temporal changes of densities or something?
Yeah, the thing is that we are focusing on woody plants.
Yeah, but they're long-lived, right?
Yeah,
so
So the thing is that they produce every year, usually once a year, they produce a new bud with flowers and leaves. Then deer come and eat upon that, and then you have to wait for the next year to produce the new flowers. So what we found is that every year this browsing level doesn't allow any regeneration, because there is this sexually regeneration. Okay? So it's a very, say, suitable indicator, because when the browser is so heavy, or even if you remove the animals, yes, they will recover, the plants will recover, or if you reduce. But only the rejected ones will start doing better than the preferred ones. So that's why so the preferred ones, if the browsing level is very high, you notice that every single new shoot, it will be completely eaten. So there is no chance of regeneration for them.
Okay, thank you. I understand.
I don't know if I answered your question.
I suppose essentially you assume that the browsing will always be constant and they're just continuously being
unless you change the density?
Yeah, okay, that makes sense, yeah. Okay.
And if you change the density, it takes a while to notice that in the field. Yeah? So it's something that we don't do every single year in every national park, because you don't notice that. We do it every 5 years approximately. So it gives some time for the vegetation to recover. Otherwise, no.
Okay. Yeah, okay. Thank you.
So from the 3 parks you presented, the one I know the best is Doñana. And for me, Doñana, one of the main herbivores are the goats. And you are not including them, or you're not presenting data here. And so I wanted to know if you know something about that. And also, more interesting, how the different herbivores have a joint effect together. So if there are indirect effects, for instance, if wild boar is facilitating their approach, or the opposite, or they are really, when there is a wild boar activity, you don't have deer around?
Yeah, very good question. I don't know if people could hear. She's asking me about the cattle, because in Doñana they have cattle and also horses. And we are counting the excrements of cattle. This, we are doing it in areas where the plots had no cattle droppings, but it's a question that we have to answer. And they are very interested in what is the sustainable effect of cattle that I cannot tell you here, because we cannot discriminate well when a plant is browsed by cattle or when a plant is browsed by any other animal. The thing is that cattle and horses are more grazers than browsers in comparison to red deer. Okay? Fallow deer and red deer are also different. Fallow deer are more grazers than browsers. So it depends on the animals. They are together and they compete, but fallow deer competes better in the ESA systems and areas dominated by herbaceous plants, and deer compete better in these browsing these communities. So we don't know the effect of cattle here, and this is the next project, I think, focusing on what are the sustainable populations of cattle or horses in the national park, taking into account the effect of the deer that already had. We are using these indicators of sustainability to tell managers, okay, do not introduce new herbivores because you already have this herbivore level, and the herbivores are necessary, of course, but adding a new herbivore is probably adding more pressure and is not really needed, maybe, unless they do somehow, they cause different ecological effects. No? Thank you.
Anyone else? Okay. I might have misunderstood, but when you were talking about watering the plants and how that helped, but then when you cut them, that also helped because the deer didn't notice them as much. But surely, when they grow back, they see them and they damage them anyway. So is it not really a solution?
Yeah. So the first solution is that watering the plant or cutting it, the ground biomass, has the same effect against drought. Okay. And then I said that it's also good because the first growing season, which is the most sensitive, it will go unnoticed. But then they will regrow, as you say, and they will, but the new growth is smaller than the whole ceiling that comes from the nursery. The ceiling that comes from the nursery is something like this, right? And then the new shoot is something like this. They will, next year, they will browse. Yes, probably, but it's 1 year that plant has been producing a longer root, reaching down there. So the important thing, the first 2 summers are very important because they need to create a very long, deep root to get the water for the rest of their life. So that's why it's very important what you do the first 2 years on the seedlings. Yeah.
Yeah, thank you very much for the presentation, very interesting. There are many enclosures in these national parks, and probably they are not the best replicate experiments, but have you compared the composition of the vegetation in fenced areas and non-fenced areas? Or
we have done that in Cabañeros National Park, in Montes de Toledo, but not in Doñana. In Doñana there are lots of exclosures, but some are open, broken, and so on. And we didn't have the chance to look at them. I don't know if someone else is looking at those exclosures that there are in many national parks. In Doñana, we have been doing that. And then we have a big change in the plant composition. There is almost 1/4 of the woody species that disappear. So many species that we think, why is it not Adenocarpus growing here? Why is it not growing Lonicera here? It's not growing because it went locally extinct by deer browsing. And this is something that we found in Montes de Toledo in these hunting estates with very heavy pressure. And we did that, it was exclosures for 30 years. So a long-term effect. And it's amazing because they are species that are locally extinct. It gives you a lot of information, these exclosures, but it's interesting to keep monitoring. Yeah. Thank you.
I just have a very curiosity question, because I just remembered. Because you said that the 2-year-old seedlings are more susceptible to drought than the 1-year-old, right? The transplanted ones. No?
No,
the other way around, no?
Yeah.
The 1-year-old, okay, never mind. Then forget my question, sorry. Then I remembered it wrong. Okay.
Claro, sí, es que.
I have a curiosity also. Do you have a long-term study of, to see if the increase of the density in the ungulates is correlated with the a lower value of vegetation index? Do you
browsing index?
Have you found any evidence of the association of these variables?
But browsing index, you mean, or with what variable?
Vegetation index and the
NDVI.
Yes.
NDVI. We are doing that in areas, but we're not manipulating densities. Areas where they have been browsed for longer periods, and then you can see the NDVI effect. But we don't have enclosures for longer periods.
No,
it's looking at the history of the vegetation in areas with higher pressure and areas with lower pressure. And this is something that we're doing in mountains in Pyrenees and so on. They have found some responses, but the NDVI is an index of greenness, of activity, which works very well in some areas, but not in some others, particularly in alpine areas, in areas where there is a lot of herbaceous plants, and in the Mediterranean evergreen communities, there are a lot of changes with the phenology, of course, and then it's more complicated. That's what we have found so far with the NDVI. But it's something that we are exploring in larger areas and using satellites. But I cannot tell you if they are working. So our impression is that they don't work as well as in alpine areas.
Because in a small scale, you can see the effects very well. Yeah. But I wonder if you look all the vegetation index in Doñana, for instance, and you take the value of density of ungulates, and you see that in the time, maybe it's not, I don't know. I wonder if
first of all, we are doing the NDVI lower of 2 meters. This is something that because it's different. You get the cover. Trees, there are trees, the trees, they are not browsed, and they are reflecting, you are obtaining the NDVI from the trees. Okay. So there's a lot of noise. And then we are using a mask to obtain the NDVI below 2 meters and compare the NDVI below 2 with the NDVI, the total NDVI, which includes the tree cover. And it's giving us different things. Tree cover is giving us mostly the shelter for the animals because they use that as a shelter. And the NDVI below 2 meters is giving us what they eat, so the browsing pressure. So it could be, it's something that we are using now, the NDVI below 2 meters. It gives you what you say, better than the overall NDVI.
Okay, thank you.
Thank you. So, thank you. So I found very interesting your clear indications to reach a sustainable level of herbivore density in national parks. So to your knowledge, are national park authorities taking actions to adjust herbivory densities?
Yes, they are.
Wonderful, congratulations.
They are, they are reducing the population. As you know, commercial hunting is prohibited in national parks. So hunters cannot go to a national park and kill animals. The rangers are controlling the populations. According to the data, so this is very useful for them because they want to know how many and what areas should be prioritized. So for them, it's a very useful tool and is relatively easy to measure. It's not as complicated as capturing animals and measuring the performance of animals. It's looking at plants and are better indicators because they are taking into account the whole community. So yes, they are doing that.
That's good news, because I think that we overall have the feeling that managers are quite insensitive to science, right? So
no, but this is, I think they are, because they notice it rapidly in the ecosystem.
But
yeah.
Great, yeah, thank you.
I don't know who was first. Go for it.
Okay, thank you. Now, my question was the same than Xavi's questions, but on the same line, how many deer they kill, for example, in Doñana? How many deer? How many wild boars? Because when I asked about that, they told me that they don't control the population, but that there are some people from, I think, Extremadura University who are killing a few of them just to sample the blood and to take some samples. But they are not actively controlling the ungulate population in Doñana.
They are. As far as I know, they are. They are killing the wild boars to reduce the population. And for instance, in Monfragüe and in Cabañeros, for instance, they capture alive more than 1,000 individuals per year, about 2,000 individuals per year. They capture them alive.
You know the numbers in Doñana?
In Doñana, I don't know the numbers of deer. Wild boar, I know there are a lot, but I cannot tell you how many because I don't remember.
But they do it mostly in Mazda.
What?
The wild boar control is mostly in Mazda.
Maybe you can answer better this question. Doñana,
in Doñana, they are killing wild boar and red deer mostly, but they don't keep track of the numbers. This is the problem. This is the thing.
But they know how many they kill. No?
If you look for the numbers, they don't have them, so they don't know.
Okay.
This is my opinion.
Mm.
I wanted to make a more general question on your personal opinion about these, I would call them projects, in which some people are making the statement that we need to reintroduce this rewilding approach, reintroduce domestic ungulates in natural areas, in many, or even wild ungulates, in many cases in fenced pens, and they are stating that it's very important for the maintenance of biodiversity, the vegetation, and blah blah, in a moment when we have probably the highest density of ungulates in places like the Iberian Peninsula in the last, would say, 3,000 years. Not before, but what's your personal opinion on this?
Yeah, that's a very interesting question. We are using these indicators precisely to say, okay, do not introduce new ungulates in these particular areas because you already have a problem of overbrowsing in some of the areas. Some people argue that the role of bisons, for instance, is different than the role played by red deer. But bisons, well, this is another talk, I think, because bisons is a different species, according to some scientists. The one is they are reintroducing, and we don't know how they compete with the deer. The effects of herbivores, it depends also if they are browsers, grazings, and so on. But right now, I think the problem is, what you said, it's overabundance of these animals. So introducing new ungulates with no planning. This happened, for instance, with Sierra Guadarrama National Park. And it was an extinct herbivore. We need herbivore. This is clear. We need to have our native herbivores. So when you reintroduce, you need to have a planning after that, because otherwise they will grow, and if with no predators and so on, it will cause some problems. But problems not only to species that are threatened, they are causing problems mostly with health, with diseases that are transmitted to humans or other wild ungulates or livestock. And then the shepherds start to complain and so on. So I think these indicators are good to say, okay, here you can or here you shouldn't. And in my opinion, in most areas of the Iberian Peninsula, there is no need to introduce herbivores. There is need of reducing or introducing predators or do something with these populations. Yeah.
Thank you very much. I don't know because it's now 2 o'clock to 3. So I think we can finish this now. And if you have any question more, you can do it off cameras. Yeah?
Thank you.
Thank you,
Maria.
much.